I am attired for summer. It is 83 degrees out, if you can believe it. Super nice out. And Scotty, I know, said he could not fathom me in a dress. This is a dress. I wear dresses. I own dresses. So there you go. New information about me. All right, let me get the chat going. How is my audio and visual? Okay, Steven says it's good. Johnny says that it is snowing there. Definitely not 83. That's rough. That's rough. We are going to frost this week. So it's not like I can get my garden in or anything. It's not time for that. But, it's a nice day. We'll take it. So Matt says that Scotty also owns and wears dresses, but that's okay. You know what? Science is fundamental as a stream fully supports you wearing whatever makes you feel pretty. I mean, if you want to wear a dress, wear a dress. If you want to wear Rico leggings, wear Rico leggings. I fully support you in your need to dress how you want. So CoraWorks, funny thing you mentioned about additional orange decoy cats. Where is that asshole? Fat Kitty has a rival. He does not like this cat. He's not a full-time cat. He's an interloper. His name is Schnitzel. And we call him Dumb Kitty because he has a very dumb face. And I don't know, he's here part of the time, but not full-time. I'm just overwhelmed by cats. It's just too many cats. Oh, God. Speaking of, some cat is barfing. Oh, Jesus Christ, cat. So that was Big Kitty throwing up. So that'll be fun for me to clean up after the stream. It's great times. It's very, if you ever thought that, maybe you were interested in the glamorous life I lead, there you go. Some cat barfing in the background. It's just highly glamorous. So anyways, tonight's topic is a very exciting one, more glamorous than cat barf. And that is model systems. So back before I was a total corporate sellout, I used to be a scientist. Like, I used to do work that meant something to the world. Now I just go to meetings. And I was a geneticist, and I worked on a model system. And I thought we would talk about what model systems are tonight, what some of the model systems are, why you use them, why you care about them. Why are we just not doing work on humans? If we want to learn about stuff that affects humans, shouldn't we do it in humans? And also, why don't we just study every animal and plant? Well, we will talk about that. So model systems are very popular in biology. And they are an animal or a plant. And today I'm only discussing animal ones. There are also plant model systems. And I'm also discussing fungal ones because I know a lot about those, where lessons from them can be applicable to other life forms. So certain processes in life are pretty ubiquitous. So anything that happens at the cellular level happens in lots and lots of things. So for instance, cell division or mitosis is something that every single eukaryote, which is an animal or plant or fungus or algae or anything else that has true chromosomes and a nucleus, that's a eukaryote. So we are eukaryotes. It's very similar in all of those. And so if you learn about the genes for mitosis in one organism, they're often what we would call homologous or the same in other organisms, and that process works the same. And it's really important to know about things like mitosis, because when mitosis is no longer regulated properly, people get cancer, because that's what cancer is. It's out of control cell division. So learning about something like mitosis is very, very important to human health. And I would argue that it's also just good to know about it, just to know about it, because it's wonderful to understand how life on Earth works. So that's where model systems come in. And the reason why certain critters have been chosen to be model systems is oftentimes just an accident. So there was maybe one group that used something as a model system, and they did some seminal experiment, or they did something that was really big, and then their grad students used it and they got famous, and their postdocs used it and they got famous, and then a community rose up, and then there were all these tools developed within that community. So, like genetics tools that everybody could use, that made it a really thriving system to use. And you can use lessons from that to draw from all life. So, I thought we would talk about some of those model systems. Some of them you've heard of before, and maybe some of you haven't. We're not talking about every model system, but just a few of the big ones. So, let's share my screen. All right, share screen. All right. Dun, dun, dun. Sharing my screen. And all right. So we will start with the OG model system. This is one you've heard of before. So this is the fruit fly, Drosophila melanogaster. This is the OG model system. So work on this began in about 1910. So around the turn of the century. And the first person who worked on this was a man named T.H. Morgan, Thomas Morgan. And it all began because he was doing some experiments with flies, and he was looking at their chromosomes. And flies actually only have 4 chromosomes. So they have sex chromosomes, X and Y. This is considered one chromosome. So sex is determined by the presence or the ratio of X to Y genes, kind of like it is in humans. Well, a little different than it is in humans, but a male is an X and a Y, and a female is an X and an X. So we would consider that one chromosome. And then they have a second, or 2, 3, and 4. And 4 is so tiny, tiny, tiny. It's the tiniest of chromosomes. So they don't have too many chromosomes. And flies have this really cool thing. They have polytene chromosomes. So fly salivary glands need to secrete a ton of proteins, which sounds super gross because it is. So because they need to secrete a ton of proteins, what they've done is in an individual cell, when it divides, it will replicate its DNA. And instead of packaging up into an all brand new cell, it will it'll just keep bunching more DNA copies together over and over and over. So you'll have one cell that has like hundreds of copies of the genome in it. And these chromosomes are called polytene chromosomes. And what's cool about them is they're very big. And so you can see them very easily. So, and because Drosophila only has 4 chromosomes and the fourth one is itty bitty 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 bitty, you can make maps of them. And so this is a squash of a polytene chromosome right here. And this was done even at the turn of the century, the turn of the last century, not this century. And they're stained with a stain called GIMSA, which is not important, but you can see different banding of the chromosomes. Okay. And you can use those bands as landmarks. So that's one reason why they were studying them, is they were doing cytogenetics with them, meaning they were just looking at what the chromosomes look like. And the thing is back then, they didn't know that chromosomes conveyed genetic information or heritable information. Like, they didn't know how the cell worked, right? And so we know that now, but back then they just thought it was a squishy bit in the middle of the cell. So, but the cool thing is Morgan, the scientist, is one day he noticed a fly that had white eyes. Aha, something's different. And so he understood Mendel's work. So Mendel is the person you probably read about in high school, who is kind of the father of genetics, and he's the one who worked with peas and worked out dominant, recessive, and uh, basic crosses, homozygotes, heterozygotes, all of that comes from Mendel's work. But Mendel did not know how that information was conveyed because they didn't know how the nucleus worked. But this guy Morgan, one day he found a fly that had white eyes and he did some crosses with it. And he determined that this gene is sex linked, which means it was always linked with this chromosome right here, the X chromosome. And so from that, he was able to determine that inheritance of a trait was correlated with getting a chromosome. So that was really seminal work. I mean, it won a Nobel Prize, one of the first Nobel Prizes. And he also had really smart graduate students who actually went on to win lots of Nobel Prizes too. So this was at Columbia University. Oh gosh, what happened there? Somebody got timed out. Oh dear. Well, it's okay, Peplin. You got timed out by Annette, but she didn't mean it. She loves you. It was an accident. Let's see if I can. I don't know how to get that back. I'm sorry. Anyways, so he had really smart graduate students too, who also went on to win Nobel Prizes for things. But because of this big tradition, all coming from Morgan, flies were kind of locked in as the OG model system because there were so many important experiments coming out of fly labs. So, a lot of the original things we learned about genetics and chromosomes all come from flies. And then they kind of had a research. I mean, this has been going on and going on and going on, but then kind of there was a big resurgence in the 80s. So, Christiane Nusslein Volhard, she's a German lady, and Eric Wieschaus, they did a big screen. And we won't go into what a screen is, but for developmental genes. So these were genes that were important in embryogenesis. And from there, they elucidated all of the Hox genes, homeobox genes. And these are genes that are important for setting up the pattern of the embryo. And this is a famous one of them. This is a mutation called antennapedia. The gene is called antennapedia. So this is a fly right here. Wild type means that there's no mutations in it. This is just a regular old fly. And you can see the fly has little antennae right here, these little set of structures here. These are for sensory. And if you have a mutation in antennapedia, and this is a dominant mutation, the antennae are transformed into legs, which is wild. I mean, it just blows my mind. So one organ is transformed into another organ. That would be like my arm being like transformed into like, I don't know, a spleen or something, or a leg, or a nose. It's a really, really big deal. Maybe my nose turned into an arm. So it's an entire transformation of that. And the cool thing is like, so we know a lot about how flies are developed. And maybe one of these days I'll do an entire talk on homeobox genes. But these genes are homologous to every other vertebrate on Earth. So every other creature or every other animal, not just vertebrates. So every other invertebrate and vertebrate, every other animal on Earth uses these same genes. And in humans and human embryogenesis, they set up the patterning of segmentation too, which is really cool. So we've learned so much about humans develop from flies, which is very, very interesting. And we're still learning about that. So we have antennapedia as a gene too. Now in humans, if you have a mutation, you don't get a transformation into another organ. That actually mutation is lethal. It's embryonic lethal. Those embryos, you probably wouldn't even know you were pregnant. So it is different than that, but it's the same genes getting turned on in the same sequence, which is really, really cool. So, and we learned all of that from flies. And, you can't do genetics with humans. Um, and why is that? Well, because to do something called forward genetics, you make a mutation and let the cell tell you what's going on. So, for instance, they made a mutation in a gene called antennapedia, and from that, they saw a phenotype, a mutant phenotype, and that's how they learned what the wild type function of antennapedia is to repress this. So the default will be antennae developing. So it's to repress the leg program. So you can't do that in humans, right? Like it is super unethical to make human mutants, and it's really unethical to do crosses. Like you can't just say like I want to cross this human and make babies and see what I have for phenotypes. Like you just can't do that. But you can do that with flies, and you can also assay hundreds and hundreds of progeny, which you cannot with humans easily, right? Because one couple probably has like 2 or 3 kids. Well, one fly couple has hundreds of kids. So it's much more amenable to genetics. That, and there are a lot of really cool tools with genetics because people have been doing genetics on flies since the early 1900s. So, there are things like genetic markers that you can use. There's constructs for making transgenics, which is combining the DNA in new ways so you can examine things. And that's how model systems work. And the other thing is you have a huge fly community. There's so many resources. They have something called Flybase, which is a website that's been around since the early 90s, a repository of genetic information and other information. The Genetic Society of America sponsors huge meetings about Drosophila genetics. So there's a good exchange of information. And that's why so many Nobel Prizes have come from studying flies. And I guess just to plug something that I'm super passionate about. This is all basic, what we would call basic scientific research. So right now I sell my soul to pharma. And pharma does a lot of research, but it is all applied research. And so it's like inventing a drug or inventing a device to treat a problem. Well, all of that stands on the shoulders of giants. And those giants are these little teeny tiny flies right here. We would be nowhere without the lessons we have learned from flies. I mean, it's really astounding. But they are not the only model system. They are only the OG model system. We have others. So this is another model system that I have done a lot of work on. This is a nematode, Caenorhabditis elegans. And as worms go, it's pretty cute. It's about 1 millimeter big at full size. You can just I'm like, I could see it with my naked eye when I was younger and my eyes were really good, but now that I'm middle aged, I don't know if I would be able to see it anymore. It's really, really tiny. So you do have to use a microscope to look at it. It's got kind of a transparent body. This is it's hermaphroditic. This is a hermaphrodite right here. It's really a simple creature. It's got a pharynx, a head. This is the gonad. This is the vulva where the eggs come out of. It has a gut. There's not much to it. But because there's not much to it, the cool thing, and so C. elegans got going as a model system in the 60s by this guy named Sidney Brenner, who started out as a really hardcore biochemist, but he wanted to develop another model system for studying embryogenesis. And these little wormy worms are easy to culture, and you can do genetics with them. There's tools available. But the most exciting things that C. elegans ever has to offer is they're exactly in an adult 976 cells. And I can't tell you how many cells are in a human body, right? Because it's different for every individual, you know? So, for instance, like Steven is like 6'3" or 4. Okay, he has more cells than Brookley does because Brookley's not that tall. But worms, 976, and we know the lineage of every single cell. So it starts out as a zygote, one cell, and then there are two cells. And we know the lineage of what every single cell turns into. So every single cell has a name. And so this is a wonderful system for developmental biology because you know what cell came from what. So you can, if something, a gene is expressed in a neuron, you know exactly which neuron. So, and the other cool thing in the 90s, and I actually used to work on this also, and there are Nobel Prizes that have come out of this. Certainly
Hey,
I was one
Kelly,
of them.
sorry to interrupt.
Yes.
We're only seeing slide 6. Like your slideshow is stuck.
That's so weird. Well, let's try again.
Okay, now I see 5. Yeah.
Huh. You know what? I think when I played the thing, it didn't share. So this is the flies.
Okay, so how many? Yeah, you have 6 total slides, right?
Yeah, I don't have many.
Okay, yeah, we see it
Did you
now.
guys see this one?
Wait, let me make sure I'm not just seeing it on StreamYard. You see it?
I
Okay,
see
that's
it.
good. All right, carry on.
Thanks for stopping me.
I want to see pictures.
Okay.
All right.
Thank you. So these are the flies. Sorry, we'll go back. I have not showed you very much. This is the flies. This is a regular wild type fly. This is a white fly. These are the polytene chromosomes. And these are the 4 chromosomes of Drosophila. There's the X and Y, 2, 3, and 4. And 4 is itty bitty bitty bitty. So this is the worm Caenorhabditis elegans. And there's not much to it. There's 976 cells. This is the gonad where the eggs develop and they get shot out here. And lots of Nobel Prizes have come from C. elegans. So some of the coolest ones are this is a great organism for studying developmental genetics. So some Nobel Prizes that processes would be apoptosis was first researched in C. elegans. So apoptosis is a problem called, or is a programmed cell death. So in development, some cells must die, and a there is a stereotyped sequence of genes that happen for that death to happen. So, for instance, your hand right here and in between each finger. So during embryogenesis, there was programmed cell death that happened to give you distinct digits. So also, it's important for regulating cells that might be cancerous. You want those cells to die, and they die through programmed cell death. Well, all of this was worked out in worms, the humble little worm. So that's exciting. Also, the genetics of aging. That also was worked out. A lot of that is worked out in worms in the 90s. And I actually worked on that for a time. I did not win a Nobel Prize, but, you know, I worked with I worked on some of those genes. And let's see what else. Oh, a process called RNAi, which is so exciting. I mean, that's using small RNAs to regulate genes, which may not make much sense to you, but is just totally, totally mind blowing. So, anyways, that's C. elegans. Caenorhabditis elegans, commonly just called C. elegans. It's a nematode, which is a roundworm. They were originally found in soil, and how they grow them is on little agar plates, like petri plates that have a lawn of E. coli on them, and they just munch, munch, munch away at the bacteria. And, they're pretty easy to work with. Genetics are not quite as sophisticated with flies because with flies, you have more parts. So, I mean, there's just not many parts to a worm, right? Like there's a head, there's a tail, there's a gut, there's a gonad. I mean, there's not much going on there. Whereas with flies, there's a lot more body parts. But very cool organism. So, hopefully this next one comes up. This is the frog. If you do not see a frog, let me know. Okay. Yes, we have the frog. So this is a frog, Xenopus laevis. It's usually just called Xenopus. And this is an African clawed frog. Now, I don't think there's anything particularly excited about this frog except they just started using it. And once one lab started using it, well, their grad students all use it, and then other people use it. And pretty soon there's a whole community around it. But a lot of biochemistry experiments were done because Xenopus has pretty big eggs, right? Frog eggs are pretty big. I mean, um, they're way bigger than human eggs, and they made extracts of them, and a lot of the biochemistry of cell division was worked out with Xenopus lavens. So this is not a super popular model system, but I mean, you'll see Xenopus in in the literature a lot, and it's a frog. So that's one. All right, here's a model system we all know and love. This is zebrafish. So this is Danio Ririo. So we would call them zebra danios in the hobby, or danios. But in science, like, everybody always refers to these as zebrafish. Like, they never call them by their scientific name. I don't know why that is. It's just what they do. So, but this is a little fishy fish right here. And the reason this was chosen in as a model system, so this got going in the 80s. Um, and what it has going for it is it's a vertebrate, so it has a backbone. So for instance, C. elegans and Drosophila, they don't have backbones, right? So a lot of this, actually, the lineage of this came from a lot of the fly people. They wanted to work on something that had a backbone. And they chose this little Danio right here because they're easy to breed, as I'm sure many of you know. I mean, I've never bred them. I don't breed fish, but they're not real hard to breed, and they were cheap, so they could get them from pet shops. And that's why they started with them. And the other cool thing they have going for them is they have these beautiful transparent embryos. And so you could watch them develop under the microscope without harming the embryo itself. So you could watch development happen in real time. And so a lot of developmental work has been done in zebrafish. We know so much about how vertebrates develop because of studies on zebrafish. And you can do genetics with them. It's a little cumbersome because, like, it's real easy to keep 100 strains of C. elegans because, it doesn't take up that much room. But it's pretty hard to keep 100 strains of fish going, as I'm sure many of you know who have fish rooms. But you can do genetics with it. So that's the zebrafish. And, we're starting to see Nobel Prizes coming out of that work too. So there's a very thriving community of zebrafish researchers. And then the last model system I'm going to tell you about is my favorite model system. So this is yeast. This is the yeast Saccharomyces cerevisiae. This is a budding yeast, and it is used to bake bread and brew beer. So it is a very, very popular yeast. This is not the yeast infection yeast. So that is Candida albicans, and they are very, very, very distantly related. So there's a lot of evolutionary daylight between them. But this is baker's yeast. So this is what I worked on. And it is the most sophisticated model system for genetics. It has more tools than even Drosophila would. And you might think, well, why? Like, it has no body parts. I mean, this is a single-celled organism. So this is a yeast here. And you see these little blobbies here. So these are buds. Yeast reproduces. It does mitosis by just producing little buds. It buds off. And it doesn't make filaments. It makes discrete sort of bodies, unless it has a mutation, which you can do. But it has the most genetic tools of anything. And there's a couple reasons for that. So first of all, you can use something called auxotrophic markers, which is a very big word. But auxotrophy means that if it has a mutation in a gene, it can't grow on media lacking it. So every strain can grow on rich media, which means it has all the goodies in it. Okay. It has tryptophan, it has serine, it has uracil. Okay. So those are all building blocks to build a yeast. Now, everything can grow on that. But if you have dropout media, it means it's lacking uracil and it has to have a gene to make it on its own. So this is very useful for geneticists because you can do screens for things by using that as a marker. So you look for things that won't grow on uracil dropout media, or you look for things that will grow on uracil dropout media. So those are oxytrophic markers, which are super useful. The other big tool that Saccharomyces has, and this is it's just a gift to the gods, and that is site-directed mutagenesis. So, if you are a geneticist and you want to make a transgenic, which means that you alter the DNA of an organism, in every other eukaryote on planet Earth, pretty much, you don't have a choice of where you insert that piece of DNA, except until recently. You don't have a choice. So it would insert randomly. But with Saccharomyces, you have site-directed mutagenesis, which means you can go in and you can replace just that gene. And that's just a gift of the gods. I mean, it's so incredibly powerful. And that's all I did for my career. So I did a lot of site-directed mutagenesis. I would make new strains every week. It's just a thing I did. Whereas in Drosophila, making a transgenic is so much more work. I mean, it could take a full year to make a transgenic, whereas I could make them like, Tuesday through Thursday, I would have one made. So it's very powerful. And the kinds of things you work on with yeast are very basic, fundamental cellular questions. So things like cell division, metabolism, um, how cells work. Because, yeast doesn't have any body parts, but mitosis is mitosis is mitosis. It works pretty much the same in every organism. So, and I worked on a process called meiosis, which is how the process of making a sex cell. And we do that too. So in humans, it makes sperm and eggs. And I worked on that with yeast. And those genes are all homologous, which means they're the same in pretty much every organism that does it. So very cool. Lots of Nobel Prizes have come from yeast, lots of them. So the most famous ones, Lee Hartwell. He used to be at Fred Hutchinson Cancer Research Center. He's a geneticist who worked on all of the genes for cell division. So all the things that controlled how cells choose to divide. So what's the signal that makes them? And that's so important for understanding how cancers work, because cancer is runaway cell division. So, yeah, so those are some model systems. So some other ones, I didn't put Arabidopsis in there. Arabidopsis is the most common plant model system, but that's a great genetic tool. It's a very tiny little weed, very popular. So that's all I have to say about model systems for now, but let's see what the chat is talking about. So, all right, now I can see you again. Hopefully you'll be able to see my face because I stopped sharing. All right, there I am again. So let's see what the chat says. So first of all, do not steal my cat, Matt. That kitty is mine, and I'll show him to you because he's over here. Matt, this is my cat. You cannot steal my cat. So kitty is mine. Okay, and this is the interloper cat. So this is Schnitzel. He's a pretty nice, squishy little cat, but he doesn't technically live here, but he might as well live here. Oh, what a good kitty. Look how dumb his little face is. Should we call him Dumb Kitty? Oh, he's got a dumb face. All right, so. Okay, so let's see where we are. Eric is here. Hi, Eric. All right, so Yellowstone Aquatic says it's snowing there too. Man, that's not good. But we had snow on Monday, so 'tis the season. So Coreworks says, I need an additional orange decoy fat kitty. I mean, that's him. Like Schnitzel he's a squishy kitty. So Matt won't know which cat to take. Please take Schnitzel. He needs a home. Scotty feels most sexy when he's wearing his fishfam.link shirt. Well then. Brookley is lurking and working. Hi, Brookley. Brookley, ponds. You and Steven need to talk about tubs together and what you're going to set up. You need to have that conversation. Cat barfing. That's my life. Why do they barf so much? Barfing cat makes a sick techno beat. I don't know what kind of techno you listen to, Steven, but that's my life there. All right, everybody's saying hi. I'm hoping Peplin has gotten out of his jail because Annette pocket modded him. It's okay. I only pocket mod. It's the only modding I do. So, All right, and there's the part where I had problems with my try. So Corework says medications you or someone you know rely upon were made via testing on zebrafish, almost guarantee. Not necessarily zebrafish. I mean, they test things on lots of things. Let's put it that way. Especially older drugs. They probably weren't tested on zebrafish, but it is a very popular model system. We know a lot because of it. Um, a lot of cool research has been done in zebrafish, and there are a lot of really cool tools available for it. So, Johnny, it was not a toad, it's a frog. It's the African clawed frog. And they're kind of like ugly and mean frogs. Like, I don't think I'd want one as a pet. So Jenna says, when you name a cat, I think it does live there. We've named all the outdoor cats, and no, they cannot all live here. Many of them don't want to, but they still have names. So Darlene got her copy of the Tub Pond Handbook today. That's so exciting. If mods want to put a link to that in chat, that's a book by Ted Coletti. The new revision of that is out. This is a book I really love. It's a book I've used. It's $20. It will change your life. It's got great pictures in it, and it's such an easy method for having a beautiful tub pond, great for breeding fish, great for growing outdoor plants, a fun addition to your garden. I really love that book. Matt, he's not your cat. Fat Kitty is mine. He is my cat. You cannot have Fat Kitty. So Nancy says Schnitzel does live with you. You don't decide these things. So Schnitzel doesn't want to live here at night. He goes and does his cat business at night, but then he just wants to stay here all day. So, I mean, I don't know if Schnitzel has commitment problems. I'm not really sure. But he's kind of a love him and leave him cat. Like he doesn't want to spend the night. So Mikim says, amazing. The actual cat from the movie Alien. Good work, Kelly. Fat Kitty does make fun noises. June says hairballs are not fun to clean up, and it's shedding season, and all these cats are shedding so goddamn much. And I try to brush them, and they like being brushed, but like it's just not enough. Like I need to vacuum the cats. So Corework says there's a cute resin barrel fountain I'm thinking about turning into a bog filter for $60 on Amazon versus $100 at the stores near us. Oh, this is very exciting. You'll have to tell me about it because I love bog filters. I think they're very fun. Okay, Steven says I'm holding out until the mug tub handbook is released. Steven, you need to put that on your on your community tab. That's so funny. I laughed so hard when Ted said that. Oh, Corwork says Schnitzel has a fight club to run. There's a lot of cats in the neighborhood. There's a lot because I live near downtown and I try to get them all spayed and neutered, which my town does has a free trap, neuter, and release program. So we're trying to get them all spayed and neutered because it's just so many cats. Just man, there's so many cats. All right. So Jenna says Kelly is Schnitzel's side chick. He goes home at night, tells his other family he's going to work during the day. Yeah, it's true. I'm his side piece. It's, someone's got to be the side chick. So CoralWorks says they sell cat vacuum brushes. I would just recommend a Furminator or something instead. Most cats don't like the noise. I can't imagine. I've heard that if you have deaf cats, they don't care. So that's an option. But none of my cats are going to put up with that. But I have yet to try the Furminator. I should get one of those this weekend and see what I think of it because what I'm doing ain't working. So much cat hair. Just so many cats. So many cats. And here I am at the bottom of the chat. Why are you guys not talking more? I mean, would you like me to talk about something? Let's see. So I am assessing my pond situation for the year. So last year I have my main pond that my friend built for me, the wooden box that has the bog filter. And I had 100, I think it's 170 gallon, maybe 6 foot galvanized steel tub. And then I had a 20-gallon black plastic tub. So next year, I think what I'm going to do, so I didn't really like the look of the black plastic. So I'm going to upgrade that with a 23-gallon galvanized steel tub. So about the same size, but I just like the look better. And I think I'm going to add a 6-foot tub. I'm not sure where I'm going to put that because my yard is like 15 by 15 feet. So I'm not exactly sure where we'll put it yet. But I think I'm going to add one more. So, and then I'm choosing like which water lilies I'll grow in them. And I ordered 2 more lotuses. So I ordered micro lotuses, 2 new varieties of them because I like the small ones. They're much more manageable. So yeah, I'm making big plans for pond season 2023. So Coralworks asks, how is the aquaponics wall? Plants growing in? I mean, yeah, it's chugging along. Like I haven't seen a lot of growth yet, but it's been like 2 weeks, 3 weeks? I mean, it's not been that long. I mean, nothing's died. I think things are chugging along, things are growing well. I mean, yeah, it was a pain in the ass to get started, but it's no work to maintain. I don't do anything to maintain it. It's just there. Just there. So, I think it's okay. All right. So I might have missed some of this stream. Can you mention a few main events which changed the way researchers work and models were made? So it's different in every model system. There's always someone who starts the party. So in the case of flies, it was T.H. Morgan out of Columbia University, and then all his grad students worked on it. And all these Nobel Prizes came out of it. And after a while, you've got a community in a model system. With yeast, I'm not sure how that got going. I mean, you would think I would know since I worked on it, but I don't. And with C. elegans, it was Sidney Brenner who decided to work on it, and it's just different. Sometimes it's one big experiment, like with flies. The sex linkage experiments were kind of what got the ball rolling, but with other model systems, it was a very conscious decision to develop a model system. So with zebrafish, it was a bunch of Drosophila people wanted a vertebrate. They were developmental biologists and they wanted to study vertebrate development. And they wanted something easy to breed, cheap, and that had transparent embryos so they could watch development happening in real time. So that's why zebrafish got going. So it's different in every system. But once it gets going, you get kind of locked into a tradition, and there's all these tools for every different model system, and there's a there's a community that rises up, and that's how these things get going. So all right, Nancy says, I do want that 6 foot stock tank, but can't decide to where to put it either. I'm not as limited with space as you are, and I have too many options. Well, Nancy, it sounds like you need more than one 6 foot tank then. I mean, because you have all these cool fish you want to breed, and I assume that you want to breed your shiners, your rainbow shiners, and maybe you want to breed some other fish. And it's really, I mean, the only time I can breed fish is in a tub because I am not a good fish breeder, but they kind of breed themselves outside. So it's very nice. So maybe you need more than one. Bex is having her dinner. Have a good dinner, Bex. So, well, anyways, we're at the bottom of the chat again, and you guys are not helping me out. So what else am I going to talk to you about? Well, we had a really good Wet Your Plants last weekend. We had Ted Coletti on. That was a lot of fun. I really enjoyed that. He actually he reached out to me, so that was very exciting because I've been such a fan of his book for years. But I don't know what we're going to do this week. We don't have a topic yet. So if you have a topic idea you want for Wet Your Plants, you could tell me here. Or if you have a topic you want for this stream, if there's something you want me to talk about, I would love to hear your ideas. I mean, some things I've wanted to talk about. I think maybe next time I stream, we're going to talk about the neurotransmitter nitrous oxide, which is crazy because did you know your body makes nitrous oxide, which is usually toxic, and it regulates blood pressure. So that's kind of cool. So that I would talk about that because Jenna wanted to talk about nitrous, or she wanted to talk about neurotransmitters. So I thought maybe we talk about that one. Yeah, let's see. Other things I wanted to talk about. I want to talk about respiration because we talked about photosynthesis, but we haven't talked about respiration yet, which is the process by which glucose is burned in the presence of oxygen to gain energy. So we respire. Every organism on earth that has mitochondria respire. Plants respire. They do respiration as well. Even though they photosynthesize, they also respire. So I thought we would maybe talk about that. But other topics, I would love to have topic ideas. So Lady Rorschach says, future idea for a talk, Kelly. We talk about the fossil record of ancestors of our modern fish species. I'm still all about a tetrapod evolution talk too. Yes, we can do that because I am a huge fan of the book Your Inner Fish. And there's also a PBS special that's like 3 or 4 parts long. It's very, very good. It's for lay people. You don't have to be a scientist to understand it. You don't have to be a scientist to understand the book, Your Inner Fish. Anybody can enjoy it. I used to teach out of it. It's one of the books that I used to teach my non-majors back when I used to teach that, and they really liked that book. And it's so Neil Shubin is a paleontologist, and he works on transitions in vertebrate evolution. So the big transition he worked on were the links between creatures that swam in the sea and creatures that walked on the land. And there is a fossil he found called Tiktaalik. I'll put that in the chat because it's spelled weird. Tiktaalik. So it's an Inuit word, and I can't remember what it means, but it's a very cool little fossil, and I think you can get a plushie of it, and I would really love a plushie Tiktaalik. So, and Jake really loved he loved the PBS special too, and he read the book too, and he really likes it. Another thing I would like to talk about is the evolution of sex, because there's a really cool book that I used to teach out of called Dr. Tatiana's Sex Advice to All Creatures, and it's by Olivia Judson. So I'll put the author's last name, and you can look her up on the Amazon. So this is a very awesome book. It's so cool. It's about different sex in the animal kingdom and the different strategies of it. And sex is a very important evolutionary force. So you guys probably know a little bit about natural selection, right? So that's survival of the fittest. So that's organisms who have the genotype that allows them to survive and reproduce the most. That's natural selection. So the best suited for their environment. But there's sexual selection, which is a very potent force for evolution. So an example of that would be like peacocks. Male peacocks have showy tails, and those give them a big sexual advantage because males that have the pretty tails get all the ladies. And so that allows them to reproduce more. But the tail is kind of a detriment for the rest of life because it's harder to fly and it's more cumbersome. It probably makes them more prone to predation. But it's offset by their big advantage of getting a mate. So there's a whole book about that. I really love this book. It's just a crazy fun read. I read it in grad school first. And then I used to teach out of it too. So that's a really good book. So Lady Rorschach, very interested in Tiktaalik, which he's a very jolly looking creature. Why wouldn't you be? So she says that I also adore that Shubin also taught and still believes, or I believe still teaches human anatomy students and teaches them the correlations between us and the rest of tetrapods. Very true. So he is at I think the University of Chicago, and every person who's a professor has to teach something. You can't just do research unless you're like a Howard Hughes funded professor. Then I think they let you out of it. You teach very little then. But he's got to work for his living because he's a paleontologist, and what he teaches human anatomy, so to doctors, which is cool. And he does do comparative anatomy between other vertebrates or fossils and humans, which is so cool because we have more in common than we have different, which is the lesson of model systems, right? So a tetrapod means an organism with 4 limbs. That would be us. We have 2 arms, 2 legs. Any creature that has 2 arms, 2 legs is also a tetrapod. So fish are not tetrapods. They do not have arms and legs, but that would include frogs, so amphibians, reptiles, birds have 4 limbs. Even whales do because they have vestigial limbs that have disappeared over time, but there's still some of them remaining. So, and those all, the genes that make a tetrapod limb are the same. The same, whether it's a mouse or a human or a frog. So it's very cool. And so he talks about some of that in his book. It's actually a field called evolutionary development, or evo-devo, which I think is a really fun term. And it's it's just studying how we're more the same than we are different. And I love that. So Lady Rorschach also needs a Tiktaalik plushie in my life. So he's a very jolly looking creature. I mean, he has kind of a jolly smile. I really like Tiktaalik. So Lady Rorschach is going to get that book. I would highly recommend it to anyone if you just want if you're a person who likes reading like science books for lay people, but you want one super entertaining and funny and about sex, which is kind of salacious. And who doesn't want to read about sex? Yeah, I highly recommend it. Dr. Tatiana's Sex Advice to All Creation. It's an excellent book. So Tiktaalik means large freshwater fish. I couldn't remember that. Excellent job. She is clearly a fan of this book. So, Lady Rorschach says the idea of looking at how our modern day species have evolved over evolutionary time would be incredible to explore. Yeah, I mean, I'm super into evolution. Um, I used to teach evolution. I'm not an evolutionary biologist by training, but I really like it. I worked in a biology department for grad school that had a very, very big emphasis on evolutionary biology. So I did a lot of work with other labs that that worked on evolution. The lab next door to us worked on the evolution of sex, so chromosomal sex, which is I worked on sex. Meiosis is sex. For a geneticist, sex is two things: meiosis, which is the process that makes sperm and eggs in humans, and fertilization. Everything that happens in the middle is not important to geneticists. It's not important. So yeah, but he works on the evolution of sex, so that was very, very cool. Yeah, and there were lots of evolutionary biologists in my grad school department, so I went to lots of seminars on evolution, and I really enjoy the topic quite a bit. So Darlene says, next, wet your plants. Talk about placement of container ponds for me, if you would. Okay, very interesting. I will keep that in mind. I don't know what we're talking about next week, but Darlene, remind us of that, and I'll answer your question because there is some thought that goes into it. All right. Mikam says, hey, how did you get a hold of my diary? Well then. Okay, Lady Rorschach says, Eustonopteron, Acanthostega, and Ichthyostega are awesome early tetrapods too. I don't know these early tetrapods. You clearly know more than I do. Maybe you should be a guest on my stream and we'll talk about it together. I don't know. Mika says, I like all knowledge on evolution. It's amazing. I agree. I think it's so cool because I just think it's very interesting to learn about what organisms share in common, which is really the lesson of model systems. We are so much more similar than we are different. Even a simple little humble yeast cell shares so many genes in common with a human. It's very cool. And that's really what studying evolution is all about. Evo Devo sounds like an amazing metal band, doesn't it? That would be an awesome metal band. Evo Devo. Maybe if someone in the Fish fam wants to start a metal band, that's a name they could use. All right, Lady R says, not related to the topic, but if you love dinosaurs, read anything by Dr. David Hone. Okay, I do love dinosaurs, and my little nephews love dinosaurs too. So maybe my little nephews would also like it. They like to read. So cool story about my nephews. I'm so proud of them. So proud. So my nephew is 9. And I told him about the science fair. I'm like, Tavo, you should. His name is Tavo. And I said, you really should be in the science fair. And he said, what I want to do, Aunt Kelly, is I want to test the pH of all the lakes. I'm so excited about this, but I'm trying not to interfere too much, right? Because I would tell him like, oh, you should test it at different times and like see how the pH changes throughout the day. And like you should test different locations and see how that changes. But I'm trying not to like insert myself too much in the process, because I want him to go on his own scientific journey. But I'm going to buy him test kits and a pH meter and get him set up for that. But so they live in Madison, Wisconsin, and they have a big limnology lab there, which is the study of lakes. And I think that maybe the limnology lab could help him, because I know if I was a grad student and a 9-year-old came in wanting help, I would be pretty excited about that. So anyways, just proud aunt moment. So proud, so proud. Okay, Steven says, I like Jurassic Park for its impeccable historic accuracy. As a geneticist, it's so hard to watch that movie. I mean, even so that I'm really old, Steven. This movie that came out when I was in 9th grade. And I remember watching it just, oh God, it hurts. Even then it hurt, but I've only seen the first one. I haven't seen any of the others. I mean, should I see them? I mean, I like Chris Pratt, but I mean, are these movies gonna piss me off? Let me know. Okay, Lady Rorschach is a Bob Backer fan. He's a total maverick in the field of vertebrate paleontology. He's kind of a nut. So like he was the guy that they would always bring on like the Tonight Show. God, I'm so old. You guys, some of you guys in chat don't even know what the Tonight Show is. Like he was on with Johnny Carson, even Jay Leno. Like he would be on all the shows, and he was the crazy paleontologist wearing the cowboy hat always. But a lot of his worldviews have been kind of debunked. So, for instance, he is convinced that dinosaurs died because they all just got like dinosaur flu or diseases. But I mean, obviously, we can't say for certain because none of us were alive back then. But there is excellent evidence supporting that it was an asteroid that wiped them out. We have very, very good evidence supporting that. So, what I'm saying is, I think he's kind of a dinosaur, but he is entertaining. Lady R says, "Oh, I would be honored as a laywoman, Kelly. Feel free to let me know if you want me to join as a guest." Yeah, maybe we'll do that. That would be pretty fun. Maybe we'll do that. I don't know when it'll happen. I never know anything. All right. Steven says dinosaur train is a close second because it shows how pteranodons used to occasionally raise T-Rex babies. I have never heard of Dinosaur Train. Was this something you read to your kids or was this a kid show? I mean, I don't have any children, so I was spared whatever this is, but it sounds really, really accurate. Lady R says the Jim Henson Company at least got Dr. Scott Sampson on board with that project. Okay, so was it Jim Henson? I've never heard of it. I've never heard of it. Lady R says, check out Nightwish's Endless Form's Most Beautiful album. Okay, I know who the band Nightwish is. I can't tell you a single one of their songs. Endless Form's Most Beautiful is a line from Darwin. It's a symphonic metal band that was greatly inspired by Darwin's on Origin of the Species. Okay, I will have to check that out. I mean, I'm not usually a modern metal fan. I'm not a fan. I'm willing to give it a shot. All right, Scotty says, lady from Cyclid Club of York has been talking about reverse respiration using seltzer water to remove algae and such from plants. I think that's what it was called, at least. Yeah. Wow, worst name ever. Yeah. Okay, so full disclosure, I have not spent a lot of time reading that guy's like 200-page online screed. I'm not really convinced that method works any better than just tap water. I also don't know the point of it necessarily. Like, why would you want to like rid yourself of snails? Like, I love snails. Snails are welcome creatures in my tank. Um, I don't know if it makes people happy, I guess, but like that website was the biggest load of pseudoscience bullshit I've ever seen in my life. I don't think that person is a credentialed biologist of any forms. If it makes people happy, I think it's fine. But like, what would reverse respiration be? Like, you can't drive that process better. So, respiration, the inputs to it are oxygen and glucose, and the outputs of it are CO2 and ultimately ATP. Okay. And ATP is an energy rich molecule that can easily be used by cells. Okay. So, reverse respiration would be like feeding in excess of ATP and CO2 and driving the process backwards. Like, it just, I don't know, this makes zero sense. Yeah. So, anyways, that was a big pseudoscience screed. And if it makes people happy, it makes people happy. But like, that was clearly not peer reviewed by anyone who was a scientist. All right, Mickam says sometimes any new knowledge is like looking at the stars. You start thinking of what and how much there is to learn. Yeah, there is so much we don't know, which is, you know, it's really exciting. I mean, we're always learning new things. So I kind of started being a scientist in the 90s, back when I was an undergrad. I started working in labs when I was 19. And I think how much has changed since then. It's really exciting how much has changed. Even the past 5 years. I think about like with cancer, things like bone cancer were a death sentence even 5 years ago. Pancreatic cancer was a death sentence 5 years ago, and now it's not. I mean, people I have 2 friends who were told that there was a 95% chance that they were not going to live because of their cancer, and they're both in remission. It's just absolutely astounding how much that field has changed in so little time. So yeah, I mean, we're always learning new things. So exciting. All right, I know, Kelly, the DNA extraction in amber was just too much. No, the rest of them are abysmal. Yeah, Jurassic Park. Okay, so there is some traces of DNA in amber, probably, but while DNA is a very, very stable molecule, okay, in that I could take a vial of DNA and wear it dry and I could leave it on my countertop for weeks and it would probably be okay, but not millions of years, okay? It's not that stable. So, and an entire genome's worth of information, it's tough times. Steven says, if you like Chris Pratt and CGI action sequences, they are decent popcorn movies. I mean, I do like Chris Pratt. I mean, he always makes a movie better. He's a good actor. He's easy on the eyes. What's not to like? Scott, you're about to trigger Kelly. I am a little triggered. I mean, if it makes people happy, it makes people happy. But man, that is a pseudoscience screed on the internet. Like, does that person have real credentials? Like, I'm suspicious. Like, I mean, I have a real ass degree from a real ass university. I don't claim to know everything, but I have real credentials. That guy. Also, wasn't he a physicist? Like, I know that I am not qualified to write 200 pages of anything about physics. Yeah. So Lady Rorschach says Backer was a bit bonkers, but he's so likable. He is, and I think it is one thing I think that is awesome about him is he got a lot of people really excited about science, and he got people excited about dinosaurs, and he got a lot of kids excited. And that's really cool. So, yeah, I know he believes in the theory of continental population exchange disease. Yeah. But you'll take him over Jack Horner any day. That's another famous paleontologist. Kind of also a nutter. We need new paleontology heroes, I think. And Neil Shubin is one of them. He gives awesome interviews. He is so good at explaining things. And the PBS special Your Inner Fish is really, really good. It's very enjoyable. Jake liked it a lot. And Jake is not a biologist. So, Darlene, not to my knowledge, but that's not saying much. What did Darlene ask about? Oh, isn't there an eclipse tonight? I don't know. I don't know anything about astronomy, Darlene. That is not something I know about. Lady R says, I swear Michael Crichton just heard of George Poinar's work on extracting insect DNA from amber and went berserk with the first Jurassic Park novel. Yeah, I read that book when it came out. I was pretty young, but it's not that hard of a book. That's kind of how Michael Crichton was. So he died in the 90s. He was pretty young when he died. He was probably my age, which makes me feel great. His books, they always have a little tiny grain grounded in reality, but a lot That's so Andromeda strain is a lot of pseudoscience weirdoness, but he wrote good stories and those stories translate well to movies. So, you know, yup, you were right. I mean, Steven knows me really, really well. I think Steven knows the things that'll trigger me. No. Scotty says, I have no plan on trying it. Was just curious your take on it. You can try it if you want. It doesn't matter. You can do whatever you want, Scotty, as long as you're not like intentionally killing your fish. Like, I'm pretty cool with it. I always advocate that if people want to do anything to treat their plants before they put them in their tank, just give them a rinse in tap water because that'll rinse away anything on the surface that maybe you don't want. It will rinse away like any like kind of rotting little nasty bits. And I think that's good enough. And that won't harm the plants either because a lot of what people do, like alum dips and stuff like that, like that's not making your plants happy. Seltzer water isn't going to make harm your plants either. Your plants will like it. The problem is, well, it's why I don't put seltzer water in my aquariums, right? All bubbles away. So, Lady R says, I'm sorry. Anyone who says that T Rex was a primary scavenger needs to get their heads examined, referring to Horner. He also married a 19-year-old when he was like almost 70. Gross. That is gross. 70-year-olds do not marry 19-year-olds. That's not cool. I'm not okay with that. Scotty says, what is the pink, red, purple looking plant behind you? It really stands out. Why, thank you. That is Alternanthera renickii rosenberg. And I really like it. I like Alternantheras. I'll put that in the chat. Alternanthera, right? I don't know how to spell this. Something like that. And it's rosen. Oh God, I can't spell this at all. Oh, so hard. Rosenberg. Oh God. Something like that. I spelled it all wrong. I'm very certain I spelled that wrong. Alternanthera, I spelled right. Yeah, you need CO2 to grow it unless you have exceptionally low pH. Maybe you can do it, but I still think that one really does require CO2. It grows pretty slow for a stem because, as you know, I don't really like stems. But that's one I do like. Alternantheras do really well for me for whatever reason. Like Ludwigias don't do that well. Like I have Ludwigia super red. It's not doing that well. So, but Alternantheras do. A good plant. Lady R says, no, DNA degrades pretty quickly over geologic time. I believe the oldest complete genomes were dated to only 2,000 years ago. Yeah, I mean, I will say that DNA is a super stable molecule, though, compared to all the other molecules. Okay, like protein, depending on the protein, will degrade anywhere from like an hour, maybe less than an hour. Maybe you can have some that last a couple weeks. Okay, those degrade really quickly. RNA degrades in anywhere from like seconds to 20 minutes, 40 minutes. I mean, DNA is pretty stable and it's that way for a reason, because it's, all of your genetic information is stored in it. So it's very important that it be a safe, stable molecule that doesn't degrade easily. But yeah, it ain't lasting that long. So Johnny says I had to take a break halfway through Jurassic World. It was so bad. Is that the one that was just an immediate sequel to Jurassic Park? So that doesn't have Chris Pratt, but came out after Jurassic Park? I don't know. I don't remember. Lady R says, get me triggered by bringing up Jack Horner. Girlfriend, you're the one who brought him up. Between the T-Rex scavenger nonsense and the Chickenosaurus proposal. Oh dear. You triggered yourself. What kind of dinosaur is Dino from the Flintstones? I don't know. Like, to me, maybe, I don't know. I don't know what kind of dinosaur he would be. I don't think he's a sauropod because he's not that big. Maybe he's a baby sauropod. I don't know. I haven't really put a lot of thought into this. I haven't really seen a lot of the Flintstones. I mean, I know the characters, but I haven't really I can't tell you what Dino sounds like. All right, Lady R says Bacher, Shubin, Paul Sereno, Philip Curry, all my heroes from childhood. Never forget Mary Anning. I read a novel on Mary Anning by the woman who wrote Girl with a Pearl Earring. I can't remember what it was called, but it's a very good book. I liked it. I read it year before last. Tracy Chevalier. That's the author. I'm going to put that in chat for you. Cannot remember the name of the book. Doesn't matter. You will use the Google. Chevalier. Anyways, it's a novel on Mary Anning. Really enjoyed it. So this lady was this was in, I'm gonna say, around 1820, and she was a woman paleontologist. She got her start as a beachcomber. She's the first person who found a complete ichthyosaurus skeleton, so many other fossils, and she never had any formal education. She came from a very poor background. She was poor her entire life, but just a legendary fossil hunter and paleontologist. Pretty cool. Nancy B says, what about Ross Geller as a paleontologist? So it was never clear to me what his specialty was because he would say that he was a dinosaur guy, but when they would show him in the museum, like they would show him in the hominid room, with like the peoply looking creatures. And I mean, there's like millions and millions and millions of years between dinosaurs and people. So, like, Ross, were you any good as a paleontologist? I don't think you were. Plus, you were spending all your time like sucking at having a girlfriend. Lady R says he was a Cenozoic expert, but the Friends writers occasionally got something right. So, Cenozoic, that's the era we're from, that hominids are from. But then they would say he was a dinosaur expert. So it was never clear to me what Ross was good for. What was Ross good for? He was, in my opinion, the most annoying friend. I don't know what the chat thinks. I think the best friend was Phoebe. I mean, she was the coolest. Carl Work says Michael Crichton wrote a bunch of good fear-mongering man versus technology stories. My favorite is The Swarm, but Andromeda Strain is a close second. I never read The Swarm. That's one of his few books I did not read. I usually think The Great Train Robbery was his best book, and that's a book where he didn't try to do any pseudoscience. So I really enjoyed that one. Um, I honestly wonder how well his books hold up. My guess is they don't hold up very well. Megan says, you want more subscribers and more debate? Perhaps make a stream on pseudoscience that were actually true. Gotta be this one thesis or two that happen to be correct in some way. Probably. I can't think of any examples right now, but there's gotta be some. I mean, we'll have to think about that. I mean, I always want some more subscribers because I'm really close to 5,000 subs. Johnny corrected my spelling on Rosenberg. That was good. I can't spell it. It's really hard to spell. It's a big word. Oh my God, Lady R, you know so many paleontologists. I don't know any paleontologists. I am not that kind of biologist. When I was in grad school, my lab mate and we used to have a joke that we would say, if it happens outside the nucleus, we just don't care. So I know very little about paleontology. Can't know everything, you know. But she knows lots of paleontologists. So the Larsons that discovered Sue the dinosaur, isn't that the T-Rex that's at Chicago Field Museum? I think I've seen that dinosaur before. It's very cool. The late Keith Rigby and John Ostrom, all awesome paleontologists. You know way more paleontologists than I do. I'm not a paleontologist, nor do I play one on TV. Johnny says they sometimes sell alternate there, Renicki Rosenberg. So, Scotty, you have a CO2 tank right now, and your water is super soft. I think it would grow pretty well from you. I don't even have super soft water. Just think you need CO2. But like Steven says, alternate theras don't do well for him. But for me, they do good. So I like them. Lady R says Nizer Ibrahim is awesome as well with his work on Spinosaurus. Again, you know roughly 50,000 times more about paleontologists than I do. I'm just not a paleontologist. You can't know everything. Scotty says I have 2 types of alternate thera. I love them. That's great. I really love them too. I used to have regular alternate thera. My Optimus Ocellaris gobies fucking shredded it. And then in my little tank, which I never showed you guys that tank, but I'll show it to you right now. Right there. My little tank. That's where my my Danios are. My what are they called? Danio Chopre. So the red plant in there is Alternanthera renickii mini, which is a plant I quite like. So it's like regular Alternanthera, but it is smaller. Focus. Focus. I don't know. Doesn't want to focus. I can't make it focus. It'll get there. All right. Lady R agrees with me that it's hardier than most. I don't know what's hardier than most. Is it Alternanthera? I don't think that's particularly hardy. It's not too. I mean, it's just a slower stem. Okay, you can't. Why cannot I think of some awesome Cenozoic paleontologists? I know they're out there. They're just not coming to mind. The only paleontologist I personally know is Chris Brochu. He's a crocodile guy. He has great talks. But you did not like Jurassic World Dominion. It just broke you. I have no clue why this won't. I don't know why my camera won't focus. But doesn't it give me like some real soft lighting? Like it kind of like smooths out my wrinkles. I mean, that's pretty good. Yeah, I never saw that movie. Maybe it's not for me though. So Lady R thinks that Dino would be some sort of sauropodomorph, would be your guess. He barks like a dog. Okay, that's good to know. We don't know what noises dinosaurs made, but the coolest thing I have recently read is they are now fairly certain that T-Rex had lips, which really makes you think about the artistic representation. I can't remember why they thought that again. I can't remember. Jake read that to me earlier, and I don't remember their justification. But anyways, yeah, had lips. So if you think about usually T-Rex, they should have like a giant mouthful of like fangs. No, had lips covering them. So pretty cool. Did they wear lipstick? Oh, Lady Art has read that book. I thought it was a really good book. I would recommend it to anyone. You don't need to be a person who likes science really to read it. It's a historical novel by Tracy Chevalier. I think she's a really good author. She wrote Girl with a Pearl Earring. It's not terribly long. You know, if you want something to read on vacation or you just want to read a nice novel. I like all her books. Okay, Darlene says that Dino is a snot chorus. Okay. Lady R says, I hate it when writers just lump paleontology into one category. I mean, it is a pretty big science, but I mean, you got to be lumped into something. Like, I'm okay being lumped into just being a biologist. Like, I'm okay with that, you know, because I am a biologist. There's lots of kinds of biologists. There's ecologists, there's geneticists, there's biochemists, there's cell biologists. I'm okay with it. I'm cool. So Coreworks says, why doesn't Ross simply eat the other friends as the largest friend? What is that from? I can't remember. Was it Futurama? Help me out, chat. I can't remember where that's from. Nancy says he was the most annoying friend. Amen, sister. Ross is the worst friend. The best friend is Phoebe. I think we can all agree with that. Lady R says, yes, Phoebe is the best friend. For me, it's a toss up between Ross and Joey for most annoying. I like Joey. I also like Chandler. But Ross, distant third among the men, last among all the friends. Of the women, I don't know if I can rank the women, except that Phoebe is the clear superior. So Nancy says, Coral, that would have been a memorable series finale. Would have. Just eat the other friends. Hi, Daniel. Darlene says, Snorkosaurus spell check corrected. Snorkosaurus. I like that. Corework says, I would argue Crichton's book aged better than the movies. Have you seen Congo? Just an awful movie. I've read the book Congo. I have never seen Congo, but I've read the book, and it is not a good book. It's not good. I think I maybe read it 9th grade. Jenna says, I think Kelly should do some reaction videos where she watches things like Jurassic Park and comments on accuracy. I mean, I could do that. I don't know how I could do that through StreamYard, but I mean, if you want to just see me bitch for 2 hours, I mean, it's a thing we could do. What's another movie that really pissed me off? Oddly enough, like Star Trek is a ton of pseudoscience. Like it's so bad, especially Next Generation. So bad. But I love it. I love it. And it's so bad. Okay, Coreworks is Nancy. It's a Futurama reference, actually. I can't take credit. I thought it was Futurama. So Nancy says that would be a fun video idea, Jenna. Triggered Kelly makes good TV. I do get triggered. But I have to say the most triggered person in chat might be Lady R. She's a little triggered right now. I mean, I get it, girlfriend. I get it. Lady R says, so sorry, Kelly. I was just a super weird kid. Kid documentaries and real documentaries, scientific papers were my jam since I was like 6. I was a nerd too, but maybe not as into paleontology. So I first thought that I wanted to be a marine biologist because, like, every single junior high girl in America goes through a phase where they either like horses a ton or they want to be a marine biologist. But friends of mine who are marine biologists tell me that it's a lot of like sorting through piles of whale poop. And that ain't what I want to do with my life. And I was never a horse girl, so I never went through that. So you gotta go through one of those two paths. Maybe modern girls don't. I mean, Steven, has your daughter ever wanted to be either a horse girl or a marine biologist? I don't know. So first I want to be a marine biologist. Then in high school, I was like, I'm going to be a botanist. And, but I also wanted to be a geneticist. So I thought I would do plant genetics. I was voted in my high school. God, this is the dorkiest thing ever. I was voted most likely to be a geneticist. That is so dorky. Just so dorky. So, yeah. And then in college, I did take a lot of botany. My degree was in biology, which is just kind of a general degree, but I took all plant classes and botany classes, so tons of them. And the way student funding worked back then is you could take more classes than your degree would require and it would still pay for them. So I took way more classes than I needed for my degree because I just wanted to take every science class. So that was my degree. And then I decided for grad school that really what floated my boat were just like basic cellular processes. So things like mitosis and meiosis and cell division and cell cycle and stuff like that. So that's why I got my PhD in that. And if I were to still be in research, I think what I would really like to work on, kind of what I'm most interested now, would be the genetics of symbiosis. So symbiosis would be like, so a famous example would be like an anemone and a clownfish. You know, they have a relationship where they both benefit. Or like the bacteria in our gut. That's a symbiotic relationship. They do things for us, we do things for them. And I'm pretty interested in the evolution of that and the genetics of it. So I think that's what I would do. But the siren call of corporate cash is so loud. I could not ignore it. And now I just go to meetings and my life has no meaning, but it has a lot of cash. So that is my career pathway. So Stephen says they only do well for me in CO2, but they can still be fussy sometimes. So Stephen is referring to alternate therapies, I believe. And I really do think they need CO2. Maybe if you have ultra low pH water, you can get away with it without it. But I still think they need CO2. Hi, Crown Tail. So Daniel Velez says Neil deGrasse Tyson is known for doing that to sci-fi movies. His podcast is great. I think Neil deGrasse Tyson is pretty cool. It always kind of gives me some nerd rage though when he tries to like be offended about things about biology because you don't really understand them that well. And it's like, just stop, just stop, just stop. Like I don't get nerd rage about physics things, I don't really understand it that well. But I think he's really cool. And I will check out his podcast. So Jake really likes whose podcast does he like? He's a physicist. And his name, I can't remember. I will ask him. But he has a podcast for lay people, and it's not just about physicists. He has different scientists on all the time. And we've listened to his podcast together at times when they had a biologist on. But yeah, that's a good podcast. Who is that? I'll tell you next time. I can't remember. But yeah, Jake is more into physics and astronomy than I am. Jake knows way more about physics than I do because I'm not that great at physics. All right. Lady R says, I just had to trigger myself by bringing up Horner. There are a few things that really trigger me, and I wanted to share with the rest of the class. Hey, we're here to support you in your time of triggering trouble. That's what this stream is all about, giving you support. Oh my God, guys. It is after 9:30. How much of this chat do we have left? Ugh, we're not even close to the bottom. We were to the bottom for a while. Oh, Lillian wants to be a teacher. That's awesome. Just like our mama was. That's so cool. Wonder what kind of teacher she will want to be. So, anyways, very cool. Chattanooga Ed, I think is streaming. I do not know. Mods, will you drop a link? I would appreciate it. Speaking of mods, thank you so much. I appreciate your modly service. I always appreciate you. Thank you everyone for coming. Replay crew, leave me a comment. I always love to see your comments. Lurkers, you have my heart. I love you too. Maybe someday you will de-lurk. I don't think I have anything else to say, but thank you all and good night.